we are going to revise on the previous class. Antibiotics are mainly either from bacteria or from fungal sources. They can be either naturally found occurring in the environment or man-made. We had studied about the origin and history, and the activity of antibiotics could be bactericidal, which means that the agent or the antibiotic will prevent the growth of the bacteria, which will keep them in a bactericidal means that the bacteria is going to be killed, and bacteriostatic means that the antibiotic will prevent the growth of the bacteria and will keep them in a stationary phase of growth. We also have spectrum. Antibiotics can be of narrow or a broad spectrum. When we talk about narrow spectrum antibiotics, it means these antibiotics are able to affect only a few number of microbes, or they can target a few types of bacteria. Broad spectrum antibiotics target many types of bacteria. So, we will move on to the different types of to the different classes of antibiotics. In the dark green region, these dark green region indicate the bactericidal agents which will cause the death of the bacteria, and the light green types or classes are bacteriostatic bacteria or antibiotics that can slow down or stop the growth of the bacteria or microbe. So we are going to deal with the first class, Beta-Lactam. Beta-Lactam class are a very common class of antibiotics. They have a central beta-lactam ring. For example, penicillin and cephalosporin are examples of beta-lactam. Examples other than these two, we have bacitracin and vancomycin, and their mode of action is to inhibit the synthesis of the bacterial cell wall. So here you have the two different beta-lactam antibiotics, cephalosporin and penicillin. Here you can observe the beta-lactam ring is present in both these two types of antibiotics. And we have covered about penicillin as it is one of the first antibiotic to be discovered. We had also studied about the uses of penicillin. And we are going to cover the mechanism of penicillin. What is the main purpose of the beta-lactam antibiotic or the penicillin, the class of antibiotics? Their mechanism is to inhibit the enzyme which is called as transpeptidase. They will inhibit the enzyme transpeptidase, which is an enzyme used for making the peptidoglycan layer of the bacteria. These antibiotics, they will disrupt the cell wall synthesis by interfering with the process of cell wall formation. Here you have, when the wall of the bacteria is formed, it is usually there is a cross linking of the peptidoglycan layer, which is composed of the NAMA. What does NAMA stand for? It stands for N-acetyl muramic acid. This red polymer is called as NAMA. And this NAMA alternates with another polymer called as NAGA or N-acetyl glucosamine. So this is the entire chain consisting of NAMA alternating with NAGA is going to be the peptidoglycan layer. The peptidoglycan layer is formed with the help of a enzyme. The enzyme is called as the transpeptidase. In this diagram, there is transpeptidase. So what will happen whenever there is a penicillin or any beta-lactam drug? It will prevent the completion of synthesis of the bacterial cell wall. See. So the transpeptidase, instead of binding to the NAGA and NAMA, it will bind with the penicillin, okay, with the beta-lactam antibiotic. And thus, when it binds with the penicillin or the beta-lactam antibiotic, it cannot participate anymore in the cell wall biosynthesis. So, if there is no cell wall, the growing bacterium will break because there is the thinning of the cell wall, which will no longer protect it from the environment. So it will break. That is the mechanism of the action of penicillin. So it leads to the formation of bacteria with a weak cell wall, which ultimately breaks up or undergoes a process known as lysis.